In this video, we'll be going through the steps involved with evaluating fixation and permeabilization for the In-Cell Western. So I'm going to open up a project here, and I'm going to open an experiment that has already been started. But just as with all other analyses in Empiria, at the beginning here, you give the analysis a name. You put in whatever kind of description that you would like up here, and then import the images from your acquisition. So next here, we're going to use a template. So there is going to be another video that focuses solely on creating templates and using the template library. This one is fairly straightforward here. We have 3 different fixation steps, either 100% methanol, 90% methanol with 10% acetone, or 80% methanol. And you can see here these are broken up with the positive and negative controls along with the background. And then each condition has its own grouping within here. So here's where you can adjust by using this little gear icon right here how the image looks on the screen. You can adjust the brightness and contrast. You can change the colors around to mix and match between red, blue, and green, grayscale, or even pseudo color if you like. Here's where you will also place the plate grid over the top of your wells so that you have everything in the correct location. You will choose your normalization channel. In this case, the cell tag 700 was used, so we're going to choose that as the normalization. This 5% trim values, what that does is that if by chance you have any of your wells that have a saturation within it, it does account for that so that that well can still be used for analysis. So we're going to create a new chart here. So we're going to look at the fold change in the 800 channel. So this is my 100% methanol, my 90% plus 10% acetone, and then 80% methanol. And these were the negative controls and the positive controls. And then you can give this chart a title if you would like. You can sort the axes. So if you want to have the 100% on the left side, and then if you want to either switch around the positive and negative controls, like this. So this gives you a chance to evaluate which blocking or which fixation step gives you the best results, the most difference between the positive and negatives, and the lowest standard deviations, which you can see would be this condition right here. So then we get into the report, and so we have the experiment name that has all the experimental conditions. You can show the template that was used if you would like to do that. And then as you scroll down through here, you have all of the data for each one of the channels individually. If you want to export any of these data, you can just click on this little export button right here and create an Excel spreadsheet with that set of data. And then down towards the bottom here, we also have the chart that is included that has the fold change included with it. And then you get a summary image of the entire experiment. Now here you can create a PDF report if you want to print it out or place it into an electronic lab notebook. You can also export this as an experiment file for somebody to review your analysis. Thank you.